Good evening, and welcome to North Adams and the Massachusetts College of Liberal Arts in the beautiful Berkshire Mountains of Western Massachusetts. Thank you all for being here in the room with us this evening, and thank you to our online audience for tuning in via the live stream. I just want to start out by thanking Jonathan Del Sordo for helping to organize everything tonight, and our tech team, Ben, to helping make sure everything runs smoothly as possible with the technology. My name's Dan Shustak. I'm one of the faculty members in the Environmental Studies Department here at MCLA. Tonight's lecture is our annual presentation of the Elizabeth and Lawrence Vadnais Environmental Issues Lecture Series. For about 20 years, we've been inviting speakers to MCLA to help us learn about and understand the important environmental issues of our day. I would like to take a moment to tell you about the people for whom this lecture series is named. Larry Vadnais came to MCLA, then called North Adams State College, to earn a Master's of Arts degree, and then several years later, in 1968, after having become a seasoned educator, he returned to North Adams State College as a professor. Then, in the 1970s and 80s, Larry worked with students to establish a radical hands-on exploration of environmental sustainability. It was called the Center for Resourceful Living. Larry's wife, Betty, along with her children, all participated in this endeavor, which included a teaching farm where students and the Vadnais family experimented with methods of sustainable agriculture and renewable energy, among other subjects. The Center for Resourceful Living profoundly changed the lives of many students. While the Center for Resourceful Living no longer exists at MCLA, today our Environmental Studies Department continues to provide a hands-on education in environmental science and sustainability, where a small community of students and faculty work closely together to understand and address those same issues that threaten the future of the planet as we know it. About 20 years ago, alumni of the Center for Resourceful Living worked with MCLA to establish the Vadnais Environmental Lecture to honor the Vadnais family and the impact of the Center for Resourceful Living, the impact that it had on their lives. Larry passed away in 2017, but his legacy continues through his former students, this lecture series, and the work of MCLA today. Now I would like to introduce one of our current students who will in turn introduce tonight's speaker. Cadence Church is a first-year student in environmental studies. Actually, she's double majoring in environmental studies and public health, a combination of majors which seems particularly relevant in light of our topic for this evening. Cadence grew up in Berlin, New York. Cadence enjoys crafting, crochet, and jewelry making, and has a particular fondness for birds. Cadence hopes to pursue graduate school and a career at the intersection of the environment and public health. Thank you for introducing our speaker tonight, Cadence.
Hello and welcome to MCLA. Our speaker tonight is Emily Monosson. Emily received her PhD from Cornell University. She spent the early part of her career working as a toxicologist. In recent years, she has focused on scientific writing, specifically writing about problems in the natural world and possible solutions to these problems. Emily is a prolific writer. She has published numerous scientific articles and several books, including Unnatural Selection: How We Are Changing Life Gene by Gene in 2014. And then in 2017, she wrote Natural Defense, Enlisting Bugs and Germs to Protect Our Food and Health. Last year, in 2023, she published her book called BLIGHT, Fungi and the Coming Pandemic. Tonight, Emily will share with us some of her insights from her latest book on fungi and how these species impact our environment and society. Tonight's presentation will be about 45 minutes, and then we will have some time for questions and answers at the end. Emily, thank you for being our speaker for this year's Vadnais Environmental Lecture Series. And MCLA, please help me welcome Emily to our campus.
Well, thank you, Cadence, and Dan and John and everybody else for being here. And it is really an honor to be part of the Vadnais Lecture Series, so I just thank you all for that. And as Cadence said, I am a toxicologist by training. And actually, the last time that I was here at MCLA was about 20 years ago, which is hard to believe. I had a course at Mount Holyoke College that was a community-based learning course, and they worked with some concerned citizens about the TCE plume. And then they came here and presented their results to the North Adams citizens. So then, since then, I've been pretty much writing books. And as Gideon said, I've been writing books about how, chemicals and how how we interact with the world and the environment and chemicals, and how that, the consequences of those interactions. And I had just finished one of those books when it was around to 2014, a little bit sooner, when late blight hit. So I don't know if anybody here grows tomatoes, but you would know that there's now this fungus-like organism that causes a disease called late blight. And if you don't know, it hit for the first time in the East Coast around 2009, I think. But then it came back again in 2014. And you have beautiful green tomatoes, and the next thing you know, they're dead almost overnight. And so I got kind of interested in this, and I wanted to learn a little bit more. And so I thought I'd write an article, and so I did some research and found out so first of all, late blight is caused by a fungus-like organism, not quite a fungus, but it's an organism that behaves very much like a fungus, and it's called Phytophthora infestans. It's the same kind of organism that was responsible in part for the potato famine, infecting potatoes in Ireland. So it's a really important pathogen. And when it hit here in the East, it was, kind of came out of nowhere. And so I talked to some scientists to try to understand where it was coming from. And it turns out that they had traced the disease to a single distributor in Florida that provided big box stores all along the East Coast with tomato starts. We all bought the tomato starts, and now we have this pathogen with us for the long haul. So that was just a sort of a personal introduction for me to what plant pathogens could do. And I'd been so focused on toxic stuff that, and around that time when that had happened, there was a paper, I was trying to think of the next book project. And so that happened. And then a paper came out by a group of scientists based in England. And it was about fungi. And they included Phytophthora infestans too, because that's often included with fungal pathogens. But it was by a group of scientists across disciplines. So not just, plant health, but also wildlife and human. And what they were doing was trying to raise awareness. And what they were saying in the paper was that, hey, look, fungi aren't just crop pathogens, and they're causing frog deaths, and they're just starting to cause deaths in bats. And they're a really important pathogen, and they don't get the respect. They don't get the respect that they really need. And so I thought, maybe that's a good topic to write about. But then it was a really depressing topic. And at the time, I thought I spoke with my editor, and it seemed like maybe I should work on a more hopeful book. So that's when I did the Bugs and Germs book, because that's kind of hopeful about how we can change our ways. And I finished that book, and the scientists who wrote the 2012 paper, meanwhile, had been just updating their papers. It's like saying, hey, listen to us. Fungi are important. They're important pathogens. We need to take them seriously. And so a newer paper came out. I saw that, and I started thinking about it. And then the CDC came out with a report of an emergent human fungal pathogen, which was a very kind of a unique thing. And so that happened in 2016, 2017, and it seemed like maybe now is the time to write that book about fungal pathogens. And so that's what I did. And I started the book before the pandemic. And, and the title came before the pandemic. And we now think about the word pandemic differently than what I was thinking back then. And actually, when I started to write it, and the pandemic happened, I asked my editors, should I still work on this? Because who's going to read a book about pandemics after this? But she said, you're halfway there. So I continued. And so a couple of themes when I was doing the research for this book that came out that were sort of through lines in the book. And one of them was transport, trade and travel. All of the fungal pathogens that I wrote about are emergent, means that they sort of emerged, they're novel, relatively novel. And many of them were caused because we moved them around. So trade and travel. And then temperature. Our internal temperature is important, as is the external temperature, climate change. So before I go into some of the details of the book, I just wanted to orient those of you who are not biology students and biologists to the word to what fungi are. So this is just the three domains of life. But we are, and all I want to point out is we are eukaryotes. We're animals. We're here. Fungi are here. Here's green plants. We all have the similar kind of cell type. We're eukaryotic cells. It means that our cell membranes are kind of similar, sort of the organelles, little organs that we have in our cells are somewhat similar, different than archaea and bacteria. Very different kinds of cell structure. And fungi are actually more closely related to us animals than they are to plants. And for many, for a long time, people had kind of grouped fungi in with plants because they look like plants, but really they're more closely related to us. And this becomes important when we're talking about pathogens. So we've got our 3 domains, and then within the domains, we've got the kingdoms of life. And so we have, plants, fungi are their own kingdom. Animalia, that is our dog, Ella. And then microbes. So. And this is one of my favorite posters, and they let you, the lab that did this poster encourages you to print it. And so if you like fungi, I recommend going to this site and printing one of these posters. But I just wanted to show, it shows the diversity of fungal life. Okay, so there are fungi, depending on who you read, millions of species of fungi, several million, some say over 10 million, but some say 3 million. So either way, it's more than animals and plants put together for diversity of fungi, if you're thinking about diversity. So it's an incredibly diverse kingdom. It includes yeasts, the bread yeast, brewer's yeast, single-celled fungi. It includes, this is one of my favorites, which I think people are probably familiar with. This is a rust. It's called cedar apple rust. And if you've ever noticed on your cedar trees in the spring, they start growing these otherworldly little jellyfish orange globs at the end. That is a rust. And what's interesting about rust is that rusts, for the most part, need 2 different hosts. So the cedar is a host and the apple tree is a host, and it uses both to complete its life cycle. So there are rusts that infect crops. This one is, cedar apple. And of course, mushrooms. We all know mushrooms. And usually when people think fungus, they think mushrooms, but it's really a diverse kingdom. And so I just wanted some characteristics of fungi that I just also want to point out that are important when we're thinking about pathogens is that most fungi produce spores. And spores is one way that fungi travel. It's one way that they live through the difficult environmental times. If they can't find a host, they're going to survive if they can make spores and their spores survive. So, and how they disperse. So, yeah, they are amazing. It's an amazing kingdom. And they're also the major decomposers. I think people probably know that by now. They're a part of our everybody has some fungus in us and on us. It's a normal part of our, microbiome. It's our mycobiome. People are probably familiar with Candida albicans. If that's a yeast. It's a yeast that causes sometimes yeast infections in various places. But it's a normal part of a biota for most of us. So these are when I wrote this book, these are sort of all of the topics that I covered. And the ones in green are the ones that I'm going to talk about tonight. So the American chestnut, the bats, and then us. So back to the CDC's announcement about Candida auris. So they issued this clinical alert in 2016, and this is a big deal. This is the CDC saying, hey, there's this emergent pathogen. We think it's really important, and healthcare workers need to take notice. They issued this in part because, one, there was an emergent fungal pathogen, which, first of all, humans, what I didn't mention, I meant to mention in the showing the diversity of all the fungi, is that the ones that are pathogens, there's millions of fungi. Pathogens are really a very small proportion of all fungi out there. It's rare to have, they're relatively rare. We're familiar with viruses and bacteria. Those are common pathogens, but fungal pathogens, there aren't that many of them. And there's a good reason for that, because most fungi don't like our warm temperature. Our body temperatures are just too warm for most fungi. They live in the environment. They tend to tolerate cooler temperatures. And so they're usually not a problem for us. So but there are some, there are a few that will infect us, like Candida albicans is a part of our bodies, but if it gets into our blood, it can be a problem. There's valley fever, which is caused by a fungus. So there are a few fungal pathogens that can tolerate our body temperatures, but most don't like it. And so this was one of the things. It was an emergent fungal pathogen. It was a yeast called Candida auris, it's a relative of Candida albicans, but it's been explained to me by one of the scientists that I spoke to at CDC that they're as different as these two yeasts are as different as we are from fish. So, just because they're yeasts and they kind of look alike, they're very different. One of the things that concerned him was that this yeast didn't seem to behave like a fungus. When people were infected, and the people who were infected tended to be hospitalized or in a long-term care facility. So they tended to be compromised in some way, often immunocompromised. And this is one of the attributes of fungal pathogens, is that they're opportunists. So if our immune system is compromised, fungal pathogens will take advantage of that. So it was in hospital settings. What was unusual is that it would spread from patient to patient and between healthcare workers and patients. Fungus doesn't usually spread like that. That was unusual. It was very difficult to disinfect the rooms. The ways that they were typically disinfecting the rooms, the hospital rooms, once a patient was gone or left the room, wasn't working for this pathogen. So there was one article in the New York Times, I think it was in 2019, about after a patient had passed away, they were actually ripping out part of the ceiling tiles and some of the wall boards because they just couldn't get rid of the fungus. So that was odd. The Candida auris was also emerged, so new, relatively new, resistant to antifungal medications, and this was a big deal. So here you have a new pathogen that for many of the strains that were infecting people couldn't be treated with some of the existing antifungal medications. So one of the reasons is, and this was one, a mystery, but two, there are only three different classes of antifungal medications. The reason for this is partly that, remember I mentioned that we're eukaryotes and fungi are eukaryotes. When you develop an antibacterial, say, to kill bacteria, what that antibacterial is doing is finding a target on the bacteria that's different from our cells, but that can stop the bacteria from growing. So they tend not to be very toxic to us because you can give somebody, or there's not a lot of side effects. You can give an antibacterial to somebody, it'll target the bacteria. We don't have what it's targeting, we're fine. Eukaryotes, more difficult to do because their cells are more like our cells, and so it's hard to find something to target that isn't going to have a lot of side effects. So there aren't that many antifungals, so for partly that reason, they're difficult to develop. Fungi also aren't a major category of pathogens, so there just hasn't been that much attention to developing antifungals. The Candida auris that was there, some of these strains were resistant to all antifungals, which meant that they couldn't even treat the patients that had it. It had a high mortality rate, upwards of 60%, which is frightening. And the strangest thing was that it seems to have emerged globally on several different continents at around the same time, which is unusual. And what I mean by this is we've been through COVID, and we all know, I think it's hard not to know that where COVID originated, that it originated somewhere in China. There is a strain of COVID that was sort of the original strain. It spread around the world, and people could track how it changed as it spread, it evolved. But we know that it can be traced back to one place, one strain, one place. So this is just sort of the, just tracing that COVID emerged here and went everywhere. But this chart, each different color, these different clades here, you don't need to I just want the visual here that if every single color was all of a sudden, this Candida auris emerged in these places. It didn't go from place to place. It wasn't one strain that emerged because of something and then spread everywhere. And that was one of the biggest mysteries about this fungus. So the question is, how would something like that happen? How would you all of a sudden have this pathogen just emerge around the world in different places, different strains? So this is where our warm bodies come in and temperature. So most fungi, like I said, don't like our warm body temperatures. this fungus called Candida auris was first discovered, I think like back in 2006. And it was in Japan, and it infected people only in the ears. So it was sort of external body parts that it could live on. But then, several years later, it was infecting people systemically. How would something like that happen, and how would it emerge all over the world? The hypothesis is that this fungus is living out in the environment, as many fungi do. It didn't really like human body temperatures. Something caused it to sort of evolve to be able to tolerate our warmer body temperature, and now people have become infected. And this is one of the, could explain why it popped up in a lot of different places at the same time, because it's an environmental pressure. And the environmental pressure would be climate change. And so I've got this fungi cast up there with Arturo Casadevall. I don't know if anybody knows his name, but he's a really interesting scientist. He's at Johns Hopkins, I think. And so he had this, he's one of the ones who sort of first put forth the idea that Candida auris might be a fungal infection, an emergent fungal pathogen that is a climate, emergent because of climate change. He also has a really interesting theory or a hypothesis that our warm body temperatures have actually, mammalian warm body temperatures, have actually can explain why, when 65 million years ago when the asteroid hit, and it was during the age of reptiles, and then the asteroid hit, and there were mammals then, but mostly reptiles. Lots of things were killed, lots of dead stuff. Fungi just had a field day because fungi are great decomposers, so there were lots of fungi. And after, when things started coming back, the mammals had warmer body temperature. The reptiles and amphibians couldn't really get up to be that warm. And so his thinking is that there was the age of mammals because the fungi weren't killing off the mammals. The fungi were infecting the reptiles and amphibians. And so that sort of enabled mammals to become the dominant, more dominant species. It's a hypothesis. So that is pretty much all that anyone knows right now about Candida auris. It's still a mystery. People are still trying to understand how possibly it would have emerged, and if this is true, that it's emergent because of climate change. And before COVID, so it was discovered before COVID, they had figured out how to disinfect rooms properly. They figured out how to control it better, and then COVID happened. Remember, it's an opportunistic. It likes people who are sick. It likes people who are immunocompromised. Once COVID hit, all those precautions, all the focus was on managing COVID. Post-COVID, Candida auris levels just rose because it took advantage of COVID, basically. COVID gave it a kind of leg up, so to speak. And so it's become more of a problem. It's spread across the states, and there's many more cases. So the next thing I want to talk about is a mammal that is also susceptible to fungi, but for a different reason. And I'm just going to read a little bit because this is sort of one of my favorite chapters, and it hits a little closer to home. And this is from a chapter called Night. For at least half a century, and probably longer, bat watching had been a summer tradition in my town of Montague, Massachusetts. The most common bat is the little brown bat, or Myotis lucificus. Myotis means mouse-eared. Lucificus means flee the light. These small bats have glossy fur and rounded ears. Just around twilight, neighbors would wander down to the old congregational church and peer up towards the second floor attic. And so this is our church. You'd go to that back corner on a summer night and you'd just see the bats streaming out. Inevitably, the bats would appear, slowly at first. A single bat would drop from the eaves, falling into the night sky before catching air. Then another, followed by 2 or 3. Moments later, they would pour from the structure into dusk. Hundreds of little brown bats leaving their roosts, taking flight, dipping, rising, and turning on a dime in search of moths, beetles, and other insects. Nocturnal hunters of an airy ecosystem. The church was a place for pregnant bats to congregate, give birth, and then nurture their young. In the spring, females would arrive at their roost pregnant, having stored sperm since mating the season in the fall, before hibernation. When the air warms and the bugs come out, mother bats go on the hunt, the newborn nursing pups sometimes clinging to them as they fly, which was something I didn't know until I wrote this book and started talking to people who study bats, which I think is kind of amazing to think about. Little browns consume about 1/3 to 1/2 of their body weight each day. Hundreds, sometimes thousands of moths and beetles will be caught and eaten, enough to provide the small mammals with a few fractions of an ounce of insects. The nutritious bits of her catch are stowed away as fat or turned into milk for her pup, while the rest she excretes as guano or bat poop. Scale up the nightly feeding frenzy from a single night to a whole season of bats wheeling overhead, and those few grams of moths, bats, beetles, and mosquitoes add up to several hundreds, possibly thousands of metric tons a year consumed by little browns across the continent. When the air chills and bugs disappear, the bats seek out a cave or mine where winter temperatures are moderated, predators are few, and there is a bit of water. These are their hibernicula. One of the largest in the Northeast is Aeola's Cave in East Dorset, Vermont, which sits slopeside in the Taconic Mountains. For decades, tens or hundreds of thousands of bats are estimated to have settled throughout Aeolus each winter. The cave is about 70 miles from Montague, and is likely that at least some of those Montague church bats overwintered there, huddled wing to wing with thousands of others, probably some from here, I would guess. A little over a decade ago, the bats began to disappear. There was a rumor that the church in Montague had cleared the attic and blocked their passage, but that wasn't the case. One by one, then in whole groups, bats were killed by innocuous-sounding fungus that causes white nose syndrome, named for the puff of white visible on their mouse-like snouts. The disease caused by the fungus Pseudogymnoascus destructans, or PD, is killing bats across North America. Killer fungal diseases are rare in mammals. Of the millions of known fungal species, only a few dozen or so are known to tolerate warm bodies, like bats or our own. Most mammal species just run too hot. We know that humans have become increasingly susceptible to infection because of the modern medical interventions that can suppress immunity. Some fungi manage to survive our warm bodies. Still, the number of disease-causing fungal pathogens that tolerate the heat remains small. We and many other mammals maintain our high body temperatures all year round, but many species of bats do not. Little browns hibernate, and when they do, their body temperatures drop. It is precisely this sort of life history loophole that white nose syndrome exploits. So bats, when they're hibernating, their temperatures will drop to match the cave temperature. So if it's 10 degrees, the bats' bodies are around 10 degrees. Since 2007, the syndrome has killed millions of bats, including little browns, big browns, northern long-eared, tricolored, and the already endangered Indiana bats. All are hibernating species. The epidemic has emptied bat caves, belfries, attics, and barns across the United States and into Canada. The losses are sobering. Pre-white nose, populations of little browns in the East numbered in the hundreds of thousands. They are now down to tens of thousands at best. Some, along with tricolored bats and northern long-eared bats, have lost 90% of their population to white nose syndrome. In 2015, northern long-eared bats were listed under the Endangered Species Act as threatened. For some of these populations, the losses are unsustainable. We are losing our bats to a fungus. And so I imagine, has anyone here noticed, over the years, the decline in bats if you had bats in your attic or wherever? So one of the really interesting things I got to do when I was researching this book was we went out batting with a I forget what you call a bat scientist, but a bat scientist. And she took this little thing that's it's like a Geiger counter almost, but for the sound. And we could just sit there and it would just make the little Geiger counter as the bats flew by you. You couldn't hear them, but you couldn't see them, but you could just hear that coming and going. And it was really kind of amazing to hear how close they come to you and you just don't even know it. So, that's a little brown bat. This is a wing of a bat infected with PD. And so all of these little spots are the fungus. The fungus is eating the keratin in the bat's wing. That's one of the things another scientist said to me when I was researching the book was, to fungus, we're just food. The fungi are just living off of the bodies of, their hosts and eating them, basically consuming them. And that is what they do. They're just making a living. And, fungus they are decomposers because they have enzymes and chemicals that they'll release that will decompose the thing they're on, which is great because otherwise the world would be piled up with dead stuff. But when they're living off of us, it's a problem. And so when these bats come out, so what happens is that the fungus is in the bat caves. When the bats go to the caves to hibernate, they'll pick up a spore or a little segment of fungus. The fungus will begin to grow in the bat. By the end of hibernation in the spring, their wings can look like this, or they can look a lot worse. When some of the photos of infected bats, their wings just look like moth-eaten sweaters. They've got holes in their wings. But the amazing thing is that if they make it through the winter period, they survive. Once their body temperature warms up, this is actually a fungus that doesn't like warm temperatures. It's how it infects the bats during the winter and the cold. And so when the bats' body temperatures warm up, they actually beat off the fungus. And so when they're found, in the summer, if they've made it through, even if their wings are beaten up, they can survive. But the thing is that they'll return to their cave. The fungi make spores. Spores can live without their host. And so they can be infected at that time. So one of the things, just like with the Candida auris, once scientists discover there's a new novel pathogen, an emergent disease, the question is, how did this happen? Where did it come from? And for this one, it's a little bit of a clearer story than something like Candida auris. The fungus was first identified in caves, Howe Caverns, which I'm guessing a lot of you out there know Howe Caverns, caves in New York. Really popular tourist attraction. And that is where white nose was first identified, was around Howe Caverns. And the scientists there were just doing their yearly assessment of bats. They go in and count bats while they're hibernating, and they saw, some dead bats. That didn't bother them too much. The next year they went back and there were dead bats everywhere littering the outside of the caves. And so this is when the alarms went out that there's something happening here. And so this right here is, kind of where Howe Caverns is. The color coding is from, the beginning, blue to, so old infections, 2006, 2007, all the way up to 2022. So you can see how this pathogen has spread throughout the country. And it's devastating bats just about everywhere, just the hibernating bats. So those that migrate aren't affected by this, although they are affected by windmills. That's like another problem, but there's something you can do about that. So the question is, where did this come from? And so when those scientists who are interested in tracking disease have looked at Pseudogymnoascus, they find it in bats in Europe. And those bats in Europe seem to be doing just fine. They're infected. They might be a little bit, impacted, but they're not dying. And so the thinking is that it's been in Europe for a long time. The bats have co-evolved to be able to live with the fungus. And tourists probably came from some cave in Europe where they were, exploring caves, and then they came to Howe Caverns and probably dropped some spores. Probably this happened several times. It probably wasn't just, person zero, but it's believed that that's sort of how this would have happened. There is some hope. So this is a relatively recent study. And, so since white nose happened, there were all these bat scientists. One of them I spoke to, John Richard, he had gotten his degree before 2006. He thought he was going to go study bats everywhere and study about their lives and ecology. And he ended up studying white nose instead. And they had been measuring bats, the weight of bats before. That's one of the things they would do, is just see how the weight of animals, it's the way you can tell how healthy they are. So they had measured, and they had been measuring bat weight, and they found that the bats that they measured the first year of white nose compared with the bats that they had measured several years later, I think around 2018 or 2017, were thinner. So the bats that were surviving later were fatter. And so they thought that maybe what happens is when white nose is infecting those bats that are hibernating, bats do wake up a little bit from hibernation. They'll get up, they'll drink a little bit of water, they'll defecate, but then they'll go back into torpor or hibernation. And so that's normal. And when bats do hibernate, they go into hibernation with just enough body fat to get them through the winter. So if they're waking, and the thought is that white nose wakes the bats up more. And if they're waking up more, they're spending more of that energy. They don't survive till the spring. And so here, the thinking is that possibly those bats that have put on more weight for whatever reason, whether it's behavioral or genetic or whatever, are the ones that are surviving. And that gives some hope that there might be some sort of genetic rescue or an evolutionary rescue possibly, so that in time, if enough of those bats survive to pass on their genes, populations will survive. I spoke to a young geneticist who was looking at the same thing about, how are some of these bats surviving? And she found, doing some genetic sequencing, that one of the genes that the bats that survive had was associated with weight gain. And so there might be a genetic component to this. It's pretty new and nobody really knows, but there's some hope that, given sort of an opportunity, the bats will survive. So there's hope. And the last story I want to talk about is chestnut, because that's another local story. Who here has seen stump sprouts? When you go hiking, you'll see these, which I never knew until I started writing this book. I had heard there were chestnuts around, but American chestnut. There are chestnuts, but American chestnut. American chestnuts were killed by a blight over 100 years ago. They're now functionally extinct. So you wouldn't think that you would find a chestnut out in the wild, but they've got very specific leaves. They've got long, thin, sort of canoe-shaped, very serrated leaves. You might see a lot of chestnuts around, but they're probably hybrids. There's Chinese chestnuts and Japanese chestnuts and all sorts of other kinds of chestnuts, and some of them are hybrids. So, but this is an American chestnut. And what happens is that those trees that were killed, from the blight over 100 years ago, died off, but the blight doesn't get into their roots. And so the stumps will just keep sprouting. And so they'll sprout, but they'll only live for about 10 or 20 years. And then blight will infect them. It causes these cankers, and the tree will just kind of fall over and die. So they don't really reach an age where they can reproduce, which is why they're called functionally extinct. And this was just an interesting story to me about how it was first, what happened when it first emerged. And it was first noticed at the New York Zoological Park, which is now the Bronx Zoo. And the Bronx Zoo, was started by the Zoological Society. It was in the late 1800s. They're going to bring these animals in from all over the world. But they had these, grounds that were just beautiful. And they had these big old trees. And they just thought that the trees were actually more important than the animals, because if an animal died, you'd replace it. But, a few 100-year-old tree dies, that was, would be catastrophic. And so they had a forester, and they hired a forester, this guy Herman Merkel, to take care of all of the plants, and he knew that park and all the plants really well. And so in 1904, Merkel was doing his rounds at the park, and he noticed a couple of American chestnut trees had leaves that looked like they were dying. But it was only a couple of trees. And so he thought, it was a tough year, there was a drought, it was too hot, it was too warm, whatever. The trees were sick, but next year they'll be okay. 1905 happens, comes back. There were something like 1,400 American chestnut trees in the zoo. All of them were infected. And he had gone, at that point, he goes to the USDA. He says, hey, what's happening with these trees? He sends them a sample, wants to know if they can diagnose it, tell them what to do. They do that. They actually misdiagnose it, and they tell them that it's a common fungus, doesn't usually infect chestnuts, and it's not usually fatal. And so they tell him what to do, which is cut off the, diseased branches and treat it with copper sulfate, which is, people still use that now to treat fungus. And he's like, this doesn't make sense. You can't treat 1,400 huge trees. And this doesn't seem like a fungus that is any kind of normal fungus. So he eventually gets another diagnosis from a colleague down the street. Turns out it's a new emergent fungus called Cryphonectria parasitica. That scientist named it something different, but it was later renamed. And subsequently, the USDA, he sent the sample to the USDA and said, this is really what it is. It's a different kind of fungus. USDA eventually identifies that fungus, again, where did it come from? So they had a program back then called Food Explorer, Agricultural Explorers, which was this program when the USDA, turn of the last century, sent men to the far-flung places of, China, Japan, Russia, wherever they could go to get plants, horticultural plants, crop plants, send them here so that we could diversify our plants, which had, double-edged sword. Most of the food we eat came from somewhere else, but all these plants came into this country, probably many of them carrying pests or pathogens with them. In this case, they found the fungus was infecting trees in China. One of their food explorers was out in China, found that there was the same fungus infecting those trees. Those trees didn't look just like the bats in Europe. Were living with the fungus. They had co-evolved. And in this case, there were Japanese chestnut trees for sale several years before the outbreak happened. So it probably came from a Japanese chestnut tree. But the finding that there was, some resistance in these Chinese chestnuts, that they were living with this disease, gave scientists an idea that maybe they could breed trees that were resistant. If they could breed American chestnut trees with Chinese chestnut trees, they could try to capture the resistance genes and breed out the characteristics that were the Chinese chestnut tree characteristics, because the two trees grow very differently, that they could maybe breed something like close to an American chestnut that was resistant to blight. They tried this in the early parts of last century without a whole lot of success. And then in 1980, a geneticist, Charles Burnham, had retired. He was an agricultural geneticist. He had grown and bred lots of different crop plants, and he thought he was going to take on this issue. He wanted to save the chestnuts. And so he developed a breeding scheme, a 30-year scheme, where they would breed American chestnuts with Chinese chestnuts, and then they would backcross them to American chestnut trees, try to breed out as much as the Chinese chestnut characteristic, and retain some resistance genes. At the time that he came up with that plan, it was believed that there were 2 to 3 resistance genes, which might not be hard to capture through breeding programs. Burnham also recognized, he was, I think, 80 when he came up with this scheme, that he wasn't going to live to see this happen. But he did know that, if he got an organization to take this on, that the program would live on. And so he founded the American Chestnut Foundation. And so this is the group that has carried on that scheme for 30 years. It's an amazing group. If you're not aware of it and you're interested in chestnuts, I suggest you look them up. They work with landowners who want to have a chestnut orchard, who want to plant some of the resistant trees, or the hopefully resistant trees that they're breeding. But what happened was, just a couple of years ago, came to the end of that 30-year period, and genetics have improved greatly over the last 30 years. And it turns out that resistance isn't just 2 to 3 genes, it's at least 12 genes spread across chromosomes, very difficult to capture. They were finding that the trees that were at the end of all this whole crossing scheme, one, weren't as American, didn't have enough of the American characteristics they had hoped, and it didn't have the resistance they had hoped. They are still working on breeding resistant American chestnuts and are very hopeful, and their goal is to breed enough that they can plant them out in forests and revive the American chestnut. At the same time, there's a program ongoing to create genetically modified chestnut trees. And so they're kind of running in tandem. And the hope is that somewhere between traditional breeding and genetic modification, eventually there will be resistant trees that can be planted back into the wild. There's that crop diseases, chestnut trees. I wrote about white pines. These are all things that were imported on, plants that were imported. And so there was a recognition back at the turn of last century that importing plants is a problem. It brings in pests and pathogens. And so for a long time, the USDA, since the turn of last century, has actually had a program where they're evaluating plants that come in. They have quarantines. They have mycologists who are looking at, what fungal pathogens there are. So this is just, I think that it's hopeful. We know that diseases still come through, but you could imagine then how many more diseases and how many more insects we'd have if we didn't have the USDA animal and plant inspection services working around the country. So they've intercepted more than 100,000 plant pests at our borders in 1 year. So this is, a few of those were fungi. Similarly, we don't have, for animals, there isn't that kind of inspection service. Livestock are inspected, but animals in the pet trade are not, which was kind of astounding to me. And the pet trade is huge. And there's also an illegal pet trade, which is huge. So animals are moving around the world bringing pests and pathogens. I didn't talk about frogs and the BD disease, but this is a fungus that's actually driven species into extinction. This is a salamander. It's a European fire salamander. There is a fungus that's infecting European salamanders in Europe called B-sal, and it's killing those salamanders. And so scientists here got very worried. We have a lot of different salamanders here. The Appalachians are actually a hotspot for salamander diversity, and so scientists who study these diseases are really concerned that this disease could come here. And so several years ago, academics working with nonprofits, working with U.S. Fish and Wildlife Service, have come up with a list of 201 salamanders that are now banned from being imported into this country because they're at high risk of bringing B-sal. And the hopeful thing is that was several years ago, and there isn't B-sal here yet. So, these kinds of things can happen, they can work. And so I just want to, to reiterate the transport, trade, and travel. We bring these fungal pathogens here. We either bring them ourselves through traveling or through trade, plants, animals. Temperature, the other theme, humans, mammals, pretty good at, preventing fungal infection because of our warm bodies. Plant crops and crops most at risk. If there's whenever I've been asked, what's the most important, fungal pathogen or what's the most scary thing, it's pathogens that infect crops, food crops. All of our food crops have major fungal pathogens that can infect them. But climate change is something that is impacting all fungal infections, from those that infect crops to those that infect us and wildlife. So I'm always asked, what can we do? That's a really hard question to answer. But I think one is to think, and that, those scientists that back in 2012 were saying, raise awareness, hey, listen, think about fungus. So, one thing we can do is just be more aware. And think about, when they tell you not to bring those bananas from one country to another, don't do it. Or, just think about I hike a lot. I've been on from here in the Appalachian Trail up towards Maine, and I do think about this, but there's not much you can do about the mud. And, just think of what you're tracking. But when you travel far places, think about the mud in your boots and maybe, clean them. But the other thing is, when we're thinking about these larger issues about, regulations and political action, these things can also help prevent new pathogens and outbreaks. So I just want to thank you all for being here, again, for listening. And, this was really, this is just the work of a lot of scientists put together in one place. And so I acknowledge them too. So I will take any questions if you have any. Anyone? Yeah. Yes, Jerry.
Are you using the concept of pandemic with respect to humans or ecosystems?
I am so glad you asked that question. So, because...
It's not a plant. It's not a plant.
Not a plant. No, I know, but I'm glad you, so I was going to have an interview one time, and I was like, oh my God, what if they asked me what I meant by pandemic? Because after COVID, we think of pandemic differently, and we think of it as, people are like, well, I've read some reviews of this, and they're like, well, there was nothing about pandemic in here, because they meant there was nothing about a human pandemic. So I looked up the word pandemic, and, pandemic means a species is impacted on a large scale in the range of its population. And so when we're talking about frogs, is certainly a pandemic. Okay, frogs around the world are being impacted by a fungus, and so that's clearly a pandemic. Could you call the white nose and bats a pandemic? Probably, because bats across our country are being impacted by a fungal pathogen. Pandemic in wheat crops and banana crops. I didn't talk about this, but bananas around the world are being infected by a fungus that is sort of a second time around. The growers just didn't learn the first time around, but. So that's how I'm using pandemic, is that it means if most of that's many different, the populations of species in a large scale are being impacted, then it's a pandemic and not just an epidemic.
Well, it also could be in the sense of a food chain and ecosystem. Eventually, our place in it still could be pandemic to us.
Yeah, that's.
If you start altering agriculture, monocultures.
Yeah. So that was, the other word I had to look up was blight, because I was like, well, does blight really apply to things that are not plant? You could say that, so. Yeah.
I noticed on your map that it seemed to be more advanced countries that were impacted by the pathogen that came all at once. And so I'm wondering how air pollution plays in.
That's a good question. I don't know. I would say that, so part of the, they're not all that advanced. I mean, we're talking, it was pretty global. One of the things is detection. So if you think of, who's finding people who have a disease, and then they're actually identifying it as that particular fungus. So you've got to have some advanced medical something to be able to do that. And one of the things is that when this did happen, the scientists at CDC, so that was, when I was writing this book, I thought I'd just go all over the place and do one of those things where you're talking to the scientists in their labs and go into the field with them, and then the pandemic hit. So I didn't get to do that, but I did get to go to the CDC, which I have total respect for the CDC. They had, they were really, there was a lot of criticism of them throughout the COVID pandemic, but when I went to the mycology branch, those people work really hard because they're getting samples from all over the world. People are like, what is this? What do we do about this? So one of the things that happened was when this was clear that this was emerging everywhere, they wondered if maybe it was something that was already out there, but just had been misdiagnosed. And because, the ability to diagnose things has improved, it keeps improving quickly. Just think about how rapidly they had a test for COVID. So through using DNA sequencing, they can do this. So they were worried that weren't worried, but they were curious to know if maybe this had been misdiagnosed, and it was out there. And it turns out that because a lot of times people archive samples, so if they get an infection, they'll just diagnose it and archive it, so they could look back and see that it actually wasn't misdiagnosed. And so it really was newly emergent. And in these countries, South Asia, East Asia, South African, maybe they were better able to diagnose it. So I don't know. That's an interesting point. I haven't seen that anywhere, but it's possible. So, and now it's everywhere. This is, the map of just the origin, but it's pretty much in most countries now. It's across most of our states. It started in the East Coast, but it's across the states. So, yeah.
It's actually going opposite the wind. So.
If people, we're bringing it. They did realize that, that hospital workers could bring it to other people, and And also, so I mentioned that fungi are opportunistic, and this one certainly is. It tends to infect people who are immunocompromised or otherwise compromised. So a lot of people might carry it too and not know they have it, and it just isn't bothering them. So, but mostly it's probably traveled with people, unlike a lot of the others that are traveling by spores and being blown by the winds. It is amazing how far some fungal pathogens can travel on the winds. Some have traveled between, across the ocean on, atmospheric winds. So, did you have a question?
The organism that causes late blight,
Mm-hmm.
you called it fungus-like.
Mm-hmm.
Can you talk more about that?
I could talk a little bit. Maybe biology teacher could talk more about it. It's an oomycete.
Well, out of curiosity, use the term Phytophthora infestans.
Mm-hmm.
It's been a while since I had my mycology course. So when I took my mycology course, that was classified as a fungus.
Okay.
And
So it's an oomycete, which is it's also called water mold. And that is more closely related to plants than it is to animals. So it's a totally different thing, but it makes spores, it spreads like a fungus, that's why it was categorized as fungus, and why it's still included when people do big reviews of fungal pathogens, they'll include the Phytophthora infestans. Also because it's so important. I mean, it infects a lot of different things. This is the other thing that's kind of more frightening sometimes about some of the fungal pathogens, is that some are very specific, like chestnut blight, Beck's American chestnut. Some of them, like BD in frogs, is affecting a lot of different species of frogs. So it's not that specific, and it can, the one that's infecting bats, lots of different species of bats. There's one called Sporothrix, which is infecting cats and humans and many other mammals. It's just whoever gets exposed to it. Because if they can live on you, they're just going to live on you. It's not like they need something really special that you have, except for maybe your keratin or whatever else they're eating. So, yeah. Any other questions? Yes.
Early in your talk, you said something about along the lines of fungi not receiving the appropriate level of respect or something to that effect. So would you make any predictions of what it would take for humans to publicly respect them?
Yeah. So in, when was it? 2022? The World Health Organization put out a list of like 19 priority fungal pathogens that had never done anything like that before. It made the news for about a week. Last of Us came out, and everybody wants to know about fungal pathogens. So sadly, what it took is popular culture, popular media. I mean, that raised awareness about fungal pathogens more than anything that, so.
well, I think you could also go back to the Irish potato famine.
Yeah.
And have that, the influence that had on the population, the shift,
Yeah.
migration just to this country,
Yeah.
pretty much based on fungus.
Yeah. Yeah. So it has social, political consequences. Actually, some people would say the similar, not about a shift, but the banana industry. So, we read articles about our Cavendish bananas, which is what we eat, are, going to be gone because of a fungus. But there are, tens of thousands of people who are employed by the banana industry, and they are impacted when a fungus hits. Right now, there is a fungus that they're having a very hard time to control. It's spread pretty much everywhere except for, like Costa Rica doesn't have it yet. That was the only other place I got to visit, and they're working very hard to keep it out. So, that's something that impacts a lot of people. And there's another fungus that a lot of people have. We eat Cavendish bananas. Those are called dessert bananas. Bananas are also a really important staple food, and a lot of people eat bananas as their staple crop in pretty much any banana growing region. And there's another fungus called black sigatoka that is impacting those bananas. And so, these are potentially really important diseases on a different scale than we think about, because we're pretty privileged, pretty much. Yeah.
Can you tell me about the Central Valley fungus?
Oh,
Affected by, because I know just a little teeny bit about that, and it sounds.
Yeah, so there's a fungus that causes something called valley fever, that one, and it had been for, typically, is a fungus that lives in the desert Southwest and areas that are like that. And I wrote just a little bit about that in the book because when I was at the CDC, they had a story of these kids in New York City that had this weird disease. They thought there was pneumonia. They couldn't figure it out. They were very sick. And it turns out that all those kids in New York, they didn't think about valley fever because these kids are in New York. But when they finally asked it, they called in the CDC, and the CDC started asking the questions. And turns out they had all traveled to, I think, someplace in Mexico where they were working on a building. And once you start stirring up the ground, those spores can be released and people can become infected. And that's one that actually can infect people who seem to be very healthy. So and it's one that scientists are concerned that climate change will increase its territory. So it won't just be confined to the desert Southwest as it is now, that it will start creeping up into other regions. And I don't want to I think it's coccidioides, but I'm not sure. I can't remember what the fungus is. But it's yeah. Does that answer your question?
Yeah, I mean, you told me more, I learned more about it, so thank you.
I mean, it infects, lungs. Yeah, it's respiratory, it begins. And it can be very hard to treat in some people because fungal infections are hard to treat because of the whole toxicity of, antifungals and resistance. And so, yeah.
Well, there are also unique types of cells that people don't really overly research, let's say. They do a lot of research on, say, bacteria, et cetera,
Yes.
and eukaryotic cells.
Yeah.
And they're eukaryotes, but the point is they're their own category, and
Yeah.
therefore they're orphans
Yeah.
in the research world.
Well, that's why they're trying to raise awareness that we need to know more about fungi than we do. So, yeah. And I think, hopefully things will change.
Thanks for coming today.
Oh, well, thank you again for turning out. Yeah, it's coccidioides. Anyway.